the remaining part of the carbohydrate metabolism. So today we'll study basically the hormonal regulation of muscle glycogen. Uh, in the last class we studied epinephrine, insulin, and glucagon, the hormones that involved in the regulation of carbohydrate metabolism. So epinephrine works in liver and muscle. That means epinephrine binds to the surface receptor of liver and muscle. Glucagon also binds to the surface receptor in liver only. In muscle, glucose enters into the glycolytic pathway, and in liver, from the glycogen, the glucose has been released, and this glucose enters into the blood. So the glucose released from glycogen will only enters into the blood from the liver, but glucose released from glycogen in the muscle will not enter into the blood. They will enter into the glycolytic pathway for energy production. So active phosphorylase enzymes increased glycogen breakdown provides fuel like glucose 1-phosphate. So how the regulation of muscle glycogen phosphorylase by covalent modification occurs? So here the phosphorylase enzyme, it has two subunits, subunit B and subunit and each subunit contains a free hydroxyl group as serine residues, and this is less active form. And glycogen phosphorylase has two forms. One is more active phosphorylase A, another is less active phosphorylase B, and which each of which contains two subunits with a specific serine residue that is phosphorylated at its hydroxyl group. These serine phosphate residues are required for maximal activity of the enzyme. So when the level of glucose reduced in the blood, or the concentration of the glycogen is sufficient in the liver, they will activate the enzyme phosphorylase B kinase. This kinase enzyme will phosphorylate phosphorylase B in this position, and now phosphorylated phosphorylase A, this activated form of phosphorylase, will be formed, and then it will trigger the breakdown of glycogen to glucose. And when the concentration of glucose is increased, then no more glycogen has to be break down. In that case, phosphorylase has to be inactivated or less activated. So there is an enzyme called phosphatase, phosphorylase A phosphatase, that change the phosphate group and convert it to the initial one, phosphorylase B, less active form. So here two enzyme kinase that catalyze the phosphorylase B phosphorylation. So based on the name of substrate, the name of the enzyme is phosphorylase B kinase, and another enzyme phosphatase that disphosphorylate phosphorylase A, and the upper name of the substrate, it is called phosphorylase A phosphatase. So liver glycogen phosphorylase is regulated by hormones and blood glucose level. Liver glycogen serve as a reservoir that release glucose into blood when the blood glucose level falls below the normal level. This glucose is released into bloodstream and carried to other tissues such as the brain that require it as a fuel. Glucose 1-phosphate formed by liver phosphorylase is converted into glucose 6-phosphate by the action of phosphoglucomutase, and then glucose 6-phosphate will convert it to glucose by phosphatase enzyme, and then this glucose will enter into the blood and responsible for increasing blood glucose level to the normal. So glycogen phosphorylase of liver as a glucose sensor. So here the phosphorylase A that is activated, and there is two allosteric site that is empty. When this enzyme facilitates the breakdown of glycogen, the glucose concentration will be raise, and then this glucose will bind in this to allosteric site, and which trigger the exposure of phosphate group like here. And then the phosphorylase A phosphatase enzyme trim this phosphate group and making the free hydroxyl group as a serine residue, and this is less active. So the activation of phosphorylase A will depend, or the activation of phosphorylase enzyme depends on the concentration of glucose. So here the regulation of glycogen breakdown during exercise or fasting. Okay, so this is basically the cascade mechanism of heparin and glucagon action. So when the concentration of glucose is reduced, then liver will synthesize glucagon, and epinephrine will be synthesized in response to lower blood glucose from adrenal cortex. So in case of lower blood glucose, glucagon will be synthesized from the pancreas, and epinephrine will be synthesized from adrenal cortex, and those two hormones will work in response to lower blood glucose level. Glucagon will work in liver, and epinephrine can work in muscle and liver. So upon binding with the cell surface receptor, they will activate GDP to GTP. GTP will activate adenylyl cyclase, which cleave ATP to cyclic AMP, and then cyclic AMP will activate protein kinase A, and protein kinase A will activate the phosphorylase kinase, and phosphorylase kinase will phosphorylate the phosphorylase B to active phosphorylase A. At the same time, the protein kinase A also deactivate glycogen synthase. So phosphorylase will cleave the glycogen or break down the glycogen to glucose 1-phosphate, and glucose 1-phosphate in muscle will enter into the glycolytic pathway, and glucose 1-phosphate in liver will be converted to glucose to produce and enter into the blood glucose. And as here glycogen breakdown is initiated, so at the same time glycogen synthesis has to be stopped. So protein kinase does that. Okay. So what is the covalent and allosteric regulation of glycogen phosphorylase in muscle and liver? This is basically same, the conversion of glycogen to glucose 1-phosphate, and then glucose 1-phosphate to glucose 6-phosphate, then 6-phosphate to glucose. So when the concentration of glucose is higher, that means the concentration of glucose 6-phosphate will be higher, and also the ATP will be higher. And then those three compounds will deactivate glycogen phosphorylase. That means the breakdown of glycogen will be stopped because there are a lot of glucose and glucose 6-phosphate. And glucose 6-phosphate will then trigger the reverse back of glucose 1-phosphate to glycogen through the activation of glycogen synthase. Similarly, in muscle, when the concentration of glucose 6-phosphate is higher, then it will stop the breakdown of glycogen. And when the concentration of glucose is higher, it will facilitate the formation of glycogen from glucose 1-phosphate. So this is the glycolysis and gluconeogenesis. So in the last slide we studied there are 4 hormones that involved in the carbohydrate metabolism regulation. That means 4 hormones that regulate the carbohydrate metabolism. One is glycogen phosphorylase, second is hexokinase, third is phosphophotokinase 1, and fifth is pyruvate kinase. So in previous slides we studied the regulation of glycogen phosphorylase. Now we'll study the regulation of the remaining 3 enzymes. So when the concentration of glucose 6-phosphate is higher, then it means there are a lot of glucose 6-phosphate. So it will stop the hexokinase enzyme, and no glucose will be converted to glucose 6-phosphate. Then glucose will be utilized for other purpose. So here the details, like hexokinase is a regulatory enzyme that catalyzes the entry of free glucose into glycolytic pathway. But whenever the concentration of glucose 6-phosphate in the cell rises above its normal level, hexokinase is temporarily and reversibly inhibited. Mammals have several forms of hexokinase, all of which catalyze the conversion of glucose into glucose 6-phosphate. The predominant hexokinase isozymes in liver is hexokinase D, and which is also called glucokinase. Similarly, when the concentration of pyruvate or concentration of acetyl-CoA is higher, then it since there is no required that no further phosphoryl pyruvate has to be converted to pyruvate. So pyruvate kinase will be stopped. So higher concentration of ATP also inhibit pyruvate kinase 8 allosterically by decreasing the affinity of the enzyme for its substrate phosphoenol pyruvate. Also, pyruvate kinase is inhibited by acetyl-CoA concentration or by higher level of long-chain fatty acid. Both of them are important fuels for citric acid cycle. Therefore, whenever the cell has higher concentration of ATP, glycolysis is inhibited by slowed action of pyruvate kinase. And when ATP concentration falls, the affinity of pyruvate kinase for phosphoenol pyruvate increases, and which enable the enzyme to catalyze the ATP synthesis, which result in a higher steady state of concentration of ATP. Okay, so this is the summary of regulation of pyruvate kinase here. Okay.